just to kind of orient everyone, I think the best way that I thought about putting this together is maybe introducing people to the technique that we've developed, orienting people a little bit on the biology, and then basically immediately after that delving into one application, then a whole host of data sets that we've kind of developed from this technique and how we've tried to better understand biology through those data sets. And then lastly, kind of summarizing questions that we haven't really been able to address that I think things like machine learning or some sort of computational modeling would really help us address and understand it. So I work in the field of directed evolution, and again, I wasn't really sure how much background was necessary, but I figured I'd go as basic as I possibly could. So what is directed evolution? So as primarily synthetic biologists or even people that are interested in maybe industrial or biomedical applications, we typically go into nature, try to find proteins that have functionalities or biomolecules that have functionalities that we're interested in. And so this is relatively, simplistically shown here where you have some sort of molecule and that gives you some specific function. But while nature is incredibly diverse and has a lot of different functionalities that we're interested in, it's not always the case that it has the one that we want. And so sometimes what we're interested in is developing or improving novel activity where we go from one arbitrary function to something that looks a little bit like it, or maybe something that's completely disparate. And so the question then becomes, how do we go from a molecule that encodes this functionality to one that encodes the novel activity or the novel functionality? And so that is, in a nutshell, the field of directed evolution. How do you apply pressure to a biomolecule to now encode some novel functionality? And it's been incredibly useful to understand biology primarily through optimization of either existing activity or the discovery of novel activities altogether. And this is, again, a very broad idea that it's not necessarily limited to catalysts or things like protein-protein interactions or even proteins at all, but extends to all biomolecules and all types of activities. And as scientists carrying out directed evolution, the problem that we're faced with is size, effectively, size and speed. In that when we start with a protein or a biomolecule, that the sequence landscape, the potential permutations of that sequence, if we're looking at mutations, even single point mutations of that gene, are pretty huge. And so this is kind of usually shown in a pictorial kind of representation like this, where there is some sort of sequence space that's plotted on the XY, where the z-axis reflects some sort of fitness. You can think of this as things like stability or activity of any sort of biomolecule. And typically what happens is most of the sequences, most of the permutations on this space are non-functional because most mutations are deleterious. But some functional sequences do occur in the course of this space, and they're restricted typically to these islands. So if you find some sort of functional variant, maybe slight permutations on that design are still relatively functional, that they don't take a huge hit in activity. But equally interesting is that these islands can occur in multiple places in this sequence space, and that even unique combinations can give rise to that same activity. And so as scientists doing directed evolution, we're concerned with primarily two pieces of information. The first is that when we pick a specific biomolecule, a specific starting activity that's somewhere on the sequence landscape, first, how do we begin to construct variations on this sequence to be able to understand the sequence space that's in the immediate vicinity of this starting point? And so each of these rings maybe describes a single point mutation. So this is the radius of one mutation, two mutations, three mutations, et cetera, that you're able to interrogate with each one of these steps. And secondly, how do we progress through the selections? How do we dictate, if this fitness level is sufficient to pass a selection, or this fitness level, or this fitness level, et cetera? And so these two ideas are basically the hallmarks of directed evolution. And simplified here, it's basically two stages: how we diversify a gene of interest, how we introduce mutations, and then how we assess those variants in some sort of functional assay. Again, you can think enzymatically things like stability, what have you. And kind of boil down even beyond that, the way that we try to assess or design some of these experiments really has two major inputs. The starting point, which is the arbitrary protein sequence that you're looking at least for us since we're interested in protein evolution, as well as some sort of substrate sequence or substrate or input activity. And we maximize both of these parameters through a whole host of adjustments, things like how efficiently we diversify, the initial state of the protein, for example, the codons that are used, or even the type of protein that we start with, the types of selections we impose, either positive and negative selection, things like neutral drift, all to maximize the probability that we'll be able to find some specific activity. And so I'll just break down both of these stages very quickly. The first one, the diversification step, is actually relatively problematic in that mutations are the drivers of Darwinian evolution, and it's how all of us in this room have come to exist today. But mutation rates that occur naturally, spontaneous mutation rates, are very, very low. So on the order of 10 to the minus 9 or 10 to the minus 10 mutants per base pair per generation. So it's a very small number. And if we just go through a very back-of-the-envelope calculation, for a generation size of about 10 billion members, which for the experiments that I'll show you later is in line with what we typically see, assuming about a 1 kb gene, that per generation you're assessing on the order of 10,000 variants. And this might seem like a lot, but for activities that are relatively complex from arbitrary starting points, you might imagine that you might need 2 mutations, 3 mutations, 4 mutations, even more. And perhaps some of those mutations occur or function epistatically in that you need to make jumps that are multiple mutations at any point in time to achieve that functionality. And so if you look at the types of coverage that you get for single point mutations of this gene, for all potential 3,000 variants, yes, you get complete and maybe slightly more than what you'd like coverage, but for anything higher than that, it's not sufficient. And so these spontaneous mutation rates are useful for evolution in a Darwinian sense in the natural world on a very, very slow time scale. But for us, we're interested in developing things on the order of months, not hundreds of thousands of years. And so we need to dramatically increase these rates to be able to assess some of these functionalities. So that's the diversification step. For the assessment strategies, there are a wide host of different techniques that have been developed for the past 3 or 4 decades that enable you to do work across a whole host of different organisms to select for a number of different parameters and activities. And this slide is just meant to highlight a couple of those in that you can incorporate things that are pretty high tech, like mass spec or HPLC to influence the selection, or you can do things that are as low tech as display mechanisms to be able to select for binding activity. But what all of these techniques, and so just to iterate, if you combine a mechanism, again, this is very simplistically, but if a mechanism to mutagenize or diversify with one of the mechanisms that's shown here or otherwise, that's basically directed evolution. But the problem with all of these schemes is that while they're fairly well worked out, these conventional mechanisms are relatively slow. So I'll walk you through kind of what a typical directed evolution experiment looks like. So you start with some gene of interest that's then introduced into some organism. And this can also happen in vitro, but typically in organisms it's a little bit easier because it does all the steps that happen downstream for you in that the functional or the genetic information is translated into some sort of functional form. So typically we don't really care about the gene, we care about the protein that's encoded by the gene, and putting that in a cell is the easiest way to have that information transfer happen. So once this occurs, we as the scientists then subject these cells to some sort of selection or screen. And what happens is we end up removing variants that are non-functional or perhaps don't even have high enough activity that we're interested in, and end up enriching for variants that have the desirable trait. Once this happens, we can then repurify this genetic information, mutagenize it to now introduce diversity onto that starting point, and reintroduce it into the cycle. And so this is the conventional directed evolution cycle. Each one of these is called a round. And typically, a conventional directed evolution report will have something on the order of 5 to 10 rounds of directed evolution. And the reason for this is that each round can take on the order of days to weeks to complete if everything is working, right? So because we're scientists, troubleshooting is the number one thing that we do, and so this can take significantly longer than this amount of time. And so while this has enabled us to understand a lot about biology and generate a lot of different biomolecules that are of interest, the problems that we can begin to address with schemes that are this slow and this labor intensive are on the easier side, in that if we want to address problems where we need hundreds of mutations to discover some novel activity, we need mechanisms to be able to survey sequence space much more efficiently, to be able to select for activities on a much, much quicker timescale.
What does the transform step refer to?
So in this case, you can approximate, this would be something like you have a plasmid with a gene on it and you put it into E. coli. Or ultimately just put it, or maybe a viral transduction that you have some genetic information on a virus and you put it in a cell.
Get it ready for expression and translation.
Basically.
Yeah.
There are mechanisms, this is maybe a little bit easier to understand, but there are mechanisms where you can do this whole process in vitro. So you have DNA and you do in vitro transcription and translation reactions and then carry out the selection that way. So in the Lou Lab, what we recognized is that all of these steps of the conventional directed evolution cycle actually already happen in biology. And for specific instances of this, it happens very, very quickly. So in the case of the M13 bacteriophage, which is a virus that infects and propagates through E. coli, that it can carry genetic information, translate it into functional form, and then result in the production of viral progeny and iterate the cycle once every 10 or so minutes. And so the fact that this undergoes this cycle in multiple orders of magnitude faster than what we do at the bench suggests that maybe this is a viable strategy to be able to do directed evolution. But what we're left with is two very important aspects, which are the hallmarks of directed evolution. How do we select for variants that are functional? And how do we introduce diversity into those variants? And so this then boils down to a typical kind of methods development for directed evolution. And we have a lot of information with regards to how to set up many of these techniques that's already available from 30, 40 years of technique development. So all we have to do is basically repurpose pre-existing mechanisms of selection or screening to better fit with this type of cycle. So what we, again, because this is very old biology, M13 bacteriophage has been studied for many decades. We know every part of how it works, all of the proteins, when they need to come on, what they do. That one specific protein kind of caught our eye as potentially providing the basis of the selection. That protein is called P3, which is encoded by the gene, also called gene 3. Old biologists were not particularly innovative in that nomenclature. So P3 carries out a number of very important functions for the life cycle of the bacteriophage. So P3 initially is responsible for recognition of the proteins that are on the surface of the bacterial cell that initiate the following cascade with respect to infection. It then undergoes a conformational change to inject the phage genome into the cells, thereby depositing all the genetic information into the cell, akin to the transformation step that I described on the previous slide. All the phage genome information is then replicated, translated into functional form, all the proteins are made, and then P3 catalyzes the process by which the nascent phage is then excised from the membrane. And so you might imagine, given all of these important functions in the life cycle of the phage, that if you remove it, which is shown here, compared to when you have P3, there's a huge differential in infectivity, right? So phage that lack P3 are basically not competent for infection and therefore not competent for propagation. And so what we thought was, given this large dynamic range and the fact that infectivity also scales with the concentration of P3 that's provided in the cell, that it would provide a suitable basis for selections downstream. And in doing so, we theoretically now solved this specific point, which is how do we incorporate selections into this mechanism? So very briefly, if we take the phage genome and remove gene 3 and encode it on a different plasmid that's inside the cell, which we call the accessory plasmid. Now, using some synthetic biology know-how, we can make selection-dependent sequences upstream of this gene that, when tripped in some way, result in the production or the transcription of gene 3 and therefore the production of P3. In place of gene 3 on the phage genome, we've now included the evolving gene of interest, and we refer to these things now as selection phage. Once the selection phage enters the cell, this is transcribed and translated into functional form, and then if it's able to trip this circuit in some way, if this activity can positively affect the selection-dependent sequence, then P3 is produced from gene 3, complements the deficiency in the selection phage, and now you have functional virus. And in doing so, this virus can then go on and infect new cells and put the genetic information in again, subject it to the selection. But all of this still leaves out one important point, is how do we introduce diversity at each one of these steps?
Yeah, it's already magic.
Yeah, biology is pretty amazing.
I just
yeah.
want to make sure I'm following.
Yeah, absolutely.
So you're saying you're not selecting for phenotype per se, you're selecting for turning on the selection-dependent sequence, which is linked in a regulatory sense to the gene 3?
Yes, so there is a phenotype, which is basically the production of variations on this which are infectious. So if you don't have this, then the rest of this capsule is made and you get virus, but the virus is non-infective. And so the phenotype ends up being the fact that the virus that is made can then go on and infect new cells and propagate through this cycle.
But I guess I'm saying you're not directly selecting for the phenotype that you're really trying to get at.
Yes, so we're using this as a conduit to inform whether or not this activity is happening in the system.
You do some programming so that when the evolving gene of interest reaches desirable configuration,
That's right.
it's more likely to trip the selection-dependent sequence.
That's right.
There's a black box there, right?
Right, and so that's kind of the synthetic biology design aspect.
Right.
And I'll show you very briefly kind of some mechanisms that we've designed to be able to link arbitrary activities of interest to that specific event.
Okay, so you're going to tell us how, like, an arbitrary phenotype that you want connects to
Exactly.
the phenotype of
Yeah.
still turning on a switch.
Yeah.
Okay.
Yeah, so I'll show you that. So right now you just have to take it on faith that this happens.
I can do
Make
that.
it so that when it does,
Just want to make sure that.
Exactly.
Okay.
But yeah, again, from like a high-level view, we still have this limitation in that, yes, we can potentially subject something to selection, but how do we introduce diversity? And what we want to do is be able to do this in a way where it directly integrates with the cycle, because then it means we as the researchers don't really have to do too much manipulation, and that way we can actually do this on the order of 10 minutes, which is akin to how it naturally happens. Yeah?
Does the host bacteria ever mutate as a receptor to which P3 binds?
To which P3 binds. Can you explain what you mean?
Does P3 need to bind to a recessive receptor?
Yes.
On the host bacteria?
Yes.
So can the host bacteria mutate the host receptor?
No. I'll also go into why that's not the case. Yeah. But that's a very good question. Okay, so what we've done now is, again, use relatively old biology, which says that there's lots of proteins that are involved in how E. coli, in this case, maintain DNA replication fidelity. It turns out then that you can make variations on those proteins that, instead of maintaining DNA replication fidelity, actually disrupt those processes. And in doing so, dramatically increase the rate of mutagenesis in the cell. And so what we've done is construct systems where we overexpress variants of known proteins that play roles in many of these different processes that are responsible for maintaining DNA replication fidelity. And when that happens, the mutation rate dramatically increases. And so in our best variant, which we call MP6, which is small molecule inducible as well, to enable us to do mutation rates across a whole very wide spectrum, that it increases the mutation rate about 320,000-fold. And if you remember the biodiversification slide, this now enables us to assess, for 1 kb gene, all quadruplet mutants per generation. So this is now much, much more useful from a directed evolution perspective. This also performs very well with strains, or comparably well to strains that have all of these processes effectively disrupted. So these are very, very sick strains. They can't really replicate DNA very well, but we can do this in a small molecule inducible way, so that's much more useful for us. And perhaps equally important to the efficiency of mutagenesis is that we also survey a very large spectrum of mutations, which we think is equally important for the process of directed evolution. So when we put all of this together, we come up with a system which we call Phage-Assisted Continuous Evolution, or PACE. And this is very simplistically how it works. Everything occurs in a constant volume vessel, which we call the lagoon. So this is effectively media at this point. We then flow in these cells that have been engineered to have a number of different properties. The first is that they encode the circuit, which will then be responsible for telling us if the activity does or does not exist, which we call the AP or accessory plasmid. They also encode these mutagenesis plasmids, which I just described, which enable us to increase the mutation rate in the system. So these cells are constantly inflowed, and we initiate a PACE experiment by seeding with selection phage that encode the gene that we want to evolve on the phage genome. What happens next is that this bacteriophage infects the cell, and the selection phage, which encodes the gene that we're evolving, is now inside the cell. And now a decision has to happen, has to be made, which is if the variant that's encoded on the SP is functional, then it trips that circuit and you make P3. We can then introduce the small molecule that upregulates the mutagenesis, mutations are accumulated in the SP, and we now have infectious progeny that also include mutations. And these then iterate this cycle, each time accumulating mutations, each time getting slightly better at tripping that circuit. But if they're non-functional, then P3 is not made, and while you will still get virus that is also mutated, these viruses actually don't go anywhere because they no longer encode for P3 and therefore can no longer infect any cells. And so to answer your question, if the receptor on the cell can actually mutate, what's happening, the most important aspect of the system is we can also control the outflow of material from this lagoon. And so we can regulate it such that the rate of outflow of everything here, cells and bacteriophage, is faster than the rate of replication of the bacterium, but slower than the rate of replication of the bacteriophage, the virus. And in doing so, any mutations that accumulate in the cells are lost, and only mutations that accumulate in functional members of the evolving gene of interest are retained in the system. And so when we crunch all the numbers compared to conventional directed evolution, we can do, instead of around every few days or weeks, we can do about 50, and this depends heavily on the selection, 50 rounds every 24 hours. And this is about a 100-fold increase in the rate of directed evolution. And because we can control the rate of outflow of material from this lagoon, the mutations effectively only accumulate in the selection phage for the SP. So this is kind of what it looks like. So the engineered cells that carry both the AP and the MP are maintained at constant density. This is all in a warm room just because it's easier to deal with. And these cells are then flowed, everything is regulated using peristaltic pumps, into these lagoons. So this is where the actual evolutions are happening. So this is a combination of media and cells that we've seeded with the bacteriophage and also induced the mutagenesis plasmid, in this case with the arabinose. And so what we do is we add phage to these lagoons, start the experiment every few hours or so, take a time point, purify the phage, and that's basically describing what happened up until that point in time during the course of the evolution. Then we can go and sequence all of that information and get potentially variants that now have improved functionality. So I'll tell you one very brief story, which was the first story that was applied to PACE, and then kind of go into some of the things that we've learned and problems that we've faced, and maybe we can start to discuss potential ways of addressing them. Yeah?
Just curious what the sort of sequence landscape was like. It just expected, I mean, I would imagine that at the beginning you'd have a very diverse pool, and then you'd start to see competition. I just wondered if that's something, if there's a kind of intuition around kind of when to stop, for example.
Yeah.
Or...
So I'm actually hoping that you guys will be able to tell me that. So at this point, I can tell you about the sequence landscape in as much as if I know what the starting point is and I know what the spectrum of mutations are, then I can predict with some reasonable efficiency what's going to happen at the first few steps with regards to the sequences that will be surveyed. After that, the math becomes relatively difficult because it's pretty combinatorial.
Like if you just sample every round, roughly, like what do you typically see?
Yeah, so what you end up seeing is a reflection of this mutation spectrum, and that tells you something about kind of the mutations that are being accumulated. But are you talking more about the rate Or I guess maybe I don't.
Well, you know, as you activate whatever this trigger is,
Yeah.
The selected phage is going to do a much better job of triggering. So
Yes.
you got to get more of that phage.
Yes.
So after 5 rounds, 10 rounds, you'll get accumulation of these selected phages.
Right.
And I just wondered if you just did next-gen sequencing over a course of several rounds.
So I have a beautiful piece of data for that I'll show you, where we basically watched. And it was the slide at the very beginning of the talk, which showed all these different genotypes kind of appearing and disappearing. So with high enough information content, we can actually describe what's happening during the course of the evolution and say something useful on that front. But yeah, I'll show a slide to that. Yeah.
Just a small method question. Is the trigger that is induced by the function sort of sensitive to how much function you have, or is it just a binary trigger and you have to later assay the protein?
Yeah, that's a very good question. So on a so there's multiple mechanisms that we can incorporate. From a design perspective, the system is able to report on variable levels of activity. Once it maxes that out, it kind of just stays there. There's no real motivation to improve. What we've done is then engineer ways to drop that bar with the continuing. So following different selections, we can make it harder, and that way it has to push up even higher. And we can do this either by stopping that experiment, manipulating the AP to make it harder, and then starting again. Or we have ways where we've basically engineered mechanisms where you add a small molecule and then the selection gets harder. And that actually we found, that'll be a hallmark, again, of one of the studies that I'll show you, that it ends up being very important, actually, to the final result, because it turns out for most things, it's very easy to find an activity. The problem is that activity is rarely as good as you want it to be. So then you have to tune a bunch of parameters, and that's really where we've learned a lot, that we can empirically now say, if we tune this knob, then the activity will do this.
If you jack up right away, then we'll never find
Exactly.
the landscape.
Exactly. So that's another really important point.
Simulated annealing, but it not
Yes. Yeah. Exactly. Yeah. So we have to step through them, and so typically what will happen is we'll design the circuit, then go into the warm room and do the experiment. And very often what will happen is the phage will disappear because everything is being constantly diluted. So if no phage is being made, then it just goes away. And so we have to make the selection easier to find some variants that do something and also introduce diversity into that starting point and then progressively make the selection harder. Right?
It's like the football field.
Oh, sorry. Yeah.
Your lagoons look to be like different sizes and stuff. Do you find that the bit of landscape that you obtain from the different
Yeah. So in that specific instance, what I was trying to do is actually test that exact question. In that, does varying the size of the lagoon actually influence, well, it's going to influence total library size, but does that influence the outcome of the selection? It turns out probably not. Realistically, you only have maybe like a tenfold window of the size of the lagoon, because below that it gets too small to really manipulate, and above that it's just too unwieldy to kind of move around. It turns out, in general, it doesn't affect it too much, but you might expect that maybe that tenfold window for really hard problems actually gives you that slight edge. Yeah?
How come your lagoon doesn't need to be shaken or stirred?
So this is actually a stir plate, and you can't see it because they're super dense, but there's a stir bar in there that's doing that. Yeah. The very first generation of this actually happened on a very large rotating kind of platform in the warm room, and then this just seemed a little bit easier because it turns out having pumps that are stationary and tubing on a rotating platform is maybe not the best idea. Okay. So the very first example that was applied to this was the evolution of T7 RNA polymerase. And the reasoning here was actually relatively simple, which is we want to connect the production the activity of a specific protein to the transcription of gene 3. And the easiest way you can achieve transcription is through an RNA polymerase. And so what, in this case, Kevin Esvelt and Jacob Carlson did was they recognized that the T7 RNA polymerase initiates very efficiently on the T7 promoter, but has no activity or very little activity on the homologous T3 promoter. And what they decided to do was evolve through a strategy that we typically use now, which is a so-called stepping stone strategy, where you design intermediates that bear hallmarks of both your starting point and your end stage. Find activity on this intermediate, and then finally transition to finding activity on the final product. And so this is schematically what I showed you in the warm room. There's two strains. The APs are slightly different. So this is the easier selection. This is the harder selection. So this is, again, going back to tuning the selection stringency. The reason this hybrid promoter even exists is because going directly to T3 was a selection that was attempted and immediately resulted in phage washout. And so what they did was start with this hybrid promoter, transition to this, carry it out in these lagoons, and you can increase the rate of mutagenesis by this inducer. This is just to kind of show you experimentally what that looked like. So we're inducing mutagenesis throughout the course of a little bit over a week, and then transitioning from an easy selection where there's lots of substrate on an easy target, then lots of substrate on a hard target, then very little substrate on a hard target. And this is what comes out, that at the beginning you start with something that's very active on the T7 promoter, no activity on the T3 promoter. As you evolve on the hybrid promoter, you find variants that now have improved activity overall, and then when you transition to what you actually care about, which is the T3 promoter, you now find variants that have this red activity, which is the T3 promoter activity. And this was done, we achieved this activity in under 108 hours. Again, compared to conventional mechanisms, where each one of these rounds would take a few days to weeks, that you'd have to carry out maybe 10 to 20 of these to be able to find all of the mutations that are actually responsible for this activity. So we've been able to apply this to a number of different proteins. And this is just a very brief summary of the ones that, at the time I made this slide, were actually functional in this context. So all of these relate the activity of a protein of interest to the production of P3 in some way. Historically, what we've done is connect them in ways where transcription is being modulated. So here, an RNA polymerase can immediately transcribe this. If we implement sort of N hybrid systems, like a bacterial 1 hybrid system or a bacterial 2 hybrid system, we can then link DNA binding activity or protein-protein interaction activity to the transcription of gene 3. We've been able to also design schemes where we liberate an inhibited RNA polymerase through proteolysis, which then links the protease activity to the transcription of gene 3, recombinases, tRNA synthetases, or even the production of bioplastics in this context. So basically, the limiting factor here is kind of twofold. Does a sensor exist for your activity of interest? And also, how much time are you willing to devote to tinkering with this to kind of develop the strategy? And this is effectively all synthetic biology. And luckily, there's a lot of circuits to report on a lot of different things. So part of that problem kind of already is solved for us. So I won't really talk too much more about the actual evolutionary kind of endpoint for the things that we've evolved. I think it's more interesting to kind of talk about what we've learned and really where we can make improvements. So I'll tell you about maybe 3 or 4 stories that we've generated to date and how each of those has kind of taught us something new, and we've tried to solve the problems that have come with each one of these different techniques. But I think potentially things like machine learning or modeling mechanisms to explain what's happening in the system would be very, very useful for us moving forward. So one of the first experiments that was done following the T7 RNA polymerase evolution story is this specific publication, which describes or attempts to interrogate the path dependency and reproducibility of evolution. So this experiment in particular has been run a number of times, but typically you need very large data sets to be able to inform a question like this. In that, if you run the same experiment over and over and over and over again, how often do you get the same answer? How often do you get a different answer? And does the way that you try to reach that answer impact how you get there? So simplistically, the way this was done is starting from the T7 RNA polymerase that initiates from the T7 promoter and then evolved along 2 separate arcs, one where it goes to the T3 promoter and then to this so-called final promoter, and another which goes through homologous but different promoter from the T3 promoter. It's called SP6, and then transition again finally to the final promoter. And you can see here's some numbers with regards to how many rounds we went through the course of this experiment. Again, you can keep in mind that number, which is the conventional number, where this would have taken decades to complete using prior ways. And what we learned here was that these things did matter, that the way that you got to the final point actually influenced the way that you evolved through these steps influenced the genotypes and also the activities that came up at the very end. But one issue that we struggled with was, how do we design these intermediates? And this is kind of maybe the highlight of one of the issues that we have, which is it's effectively an art at this point, in that we design variations, we go into the lab, we test them, that tells us something about whether or not we can actually evolve activity towards those variants, and then we iterate this design. But if we had a way to be able to interrogate things like homologs of the T7 RNA polymerase, or even inform through coevolution of promoters and RNA polymerases, perhaps we could better use that information to pick sequences that might be amenable to evolution in this context. Another issue that we kind of saw was that when we designed some of these sequences, we found that some of them actually interacted with host machinery. So there's a lot of things that interact with DNA. Many of those things end up being polymerases. So you have host polymerases that interact with some of these sequences, resulting in the production of P3 in the absence of T7 RNA polymerase. So the selection breaks down. Or things that have secondary structure where the sequence is just, once it's melted, completely inaccessible to the RNA polymerase. So these types of problems, we discover after the fact, but it would be very useful for us to be able to determine these kind of computationally or in silico before we even have to do any of these experiments. From the same data set, we observed a number of very interesting biological consequences. One of them was epistasis. So this is highlighting some of the mutations that appeared. So if you look at this schematic, each one of these experiments was done in quadruplicate along these separate arcs. And so these are the 4 populations, and then these are the 2 arcs that were shown on the previous slide. In one specific population, we observe this specific mutation, phenylalanine 646 to leucine, in only one population out of the 8. The expectation might be that we would see it maybe in this pathway, but not in this pathway. But in reality, it's actually completely unavailable in any of the other experiments that we did. And so this was one of the reasons why this whole line of experimentation was attempted. Is evolution reproducible? And this strongly suggests that maybe it's not. But beyond that, this specific mutation occurs in this lagoon in a way that's very special to mutations that existed prior to that mutation coming up. In the sense that in this specific variant, it's beneficial. In every other context across these 7 other lagoons, it's detrimental. And when we go in and look at structural information and other mutations that were accumulated in lagoon 3 of the SP6 pathway, we can then explain it by epistatic relationships that happen between accumulation of this mutation and other amino acids that surround that specific
unit. So you can specifically explain it by the 3D knowledge of the 3D conformation?
In a sense, in that you can go back and use that information to inform whether or not you can make this mutation beneficial in a slightly different context. In that if you now modify additional mutations, now the mutation is tolerated.
Could you also use the sort of coevolution structure, the covariance,
yes,
to predict?
Yes. So that's another thing that once you... Exactly. Yeah. So if the data is sufficient and you have enough time points to basically say, at this time point, both of these mutations came up, that tells you something about maybe the requirement for the secondary mutation for that mutation occurring. And that turns out to be the case here. But all of these things kind of happened in very slow sequence. And so if we had all of the sequencing information, in addition to structural information or potentially mechanisms of designing this or informing this in silico, maybe we could predict this kind of a priori.
Qualitative.
Exactly. Exactly. Okay, so this was all done with the T7 RNA polymerase story. I'll switch gears now to a slightly different problem. So this is the directed evolution of proteases, which is conceptually a much more difficult problem, and actually historically many people have attempted to address this by evolving proteases that have different substrate specificity primarily with the goal of applying them as therapeutics. Because you might imagine, if you think along the lines of antibodies, they're very good at blocking things, but you have to apply antibodies in a one-to-one mixture to the target that you're trying to inhibit. But now if you had proteases, those would be catalytic and would turn over your target much more quickly, and in that way maybe provide a basis for therapeutics. But they've been incredibly difficult to engineer, and what we found is that with PACE, actually, because of the number of rounds of directed evolution that we progress through and how many mutations we accumulate in each one of those rounds, we can actually progress in a very meaningful way to variants of proteases that now have different activities. And so in this specific story, this was work led by Michael Pecker in the Lou Lab. He wanted to evolve TEV, which is the protease that we all use for all kinds of things in the lab, from cleaving this canonical sequence to a substrate that looks nothing like the starting point. The reason TEV was chosen here is because it's very well characterized, very well behaved, and very specific. And so we're now faced with a conundrum of how we design the stepping stones to be able to evolve activity on this specific substrate. And this is what he did. And it's overwhelming because this is all the stuff that worked. In that he tried different permutations of different amino acids in different orders to try and find conditions that were permissible for evolution in this context. And this is problematic because if we have to do this for every single target, one, that's very time and labor intensive, but two, doesn't really help us understand why things do or don't work. So if we could integrate mechanisms of saying, specific targets that we want to go after are actually accessible to protease because they're structurally disordered or very flexible, that might help us. In addition, the selection of the desired target as a function of the starting point is actually very important because it influences how many different stages or rounds of directed evolution that you have to do to actually find that activity. And so finding variants that are maybe enzymes that are closer to this than TEV in this case would also be incredibly useful. Another thing that was relatively interesting that came out of this study is that what Michael found is that introducing degeneracy at specific positions that are informed by structural information between the protease and the substrate actually dramatically enhance how well these selections work. So for example, you can see here, he's targeting the first position, which is this glutamic acid to a histidine, that he randomized using NNK site saturation mutagenesis, these three amino acids to be able to target that position. And he picked those three amino acids because those are very close to that glutamic acid in the crystal structure. And so if we could perhaps think of better ways to be able to target some of these mutations, that we could dramatically enhance the efficiency of this mechanism. So now switching gears yet again, but maybe in a much more global way. So I've told you this is simplistically how PACE works. The selection phage encodes an evolving gene of interest alongside these phage genomes, and the mutagenesis plasmid overexpresses a number of mutagenic proteins to then affect mutations in the evolving gene of interest. We know something useful about the mutagenesis in that the spectrum is fairly defined. But for any arbitrary starting point, so here's the 7-mer of one protein that I use fairly regularly, you can see these are the codons, these are amino acids that are described by those codons. But is that the best starting point? Because there might be a number of different genes, all of which have a number of different codon assignments, all of which make the same protein, that might give you different starting points based on this mutation spectrum to evolve different activities. So because of this type of recoding, all of these sequences theoretically do not evolve in the same way or do not access the same sequence space for each one of these positions. And so if we could reconstruct, or sorry, not reconstruct, but rather design variations on the potential codon assignment in different genes to better inform the starting point for directed evolution, that would maximize the diversity and the discovery efforts that we're trying to lead. So this is kind of my favorite story, largely because I led it, but also because the information that came out was very interesting to see. So this, I think, addresses maybe your question with regards to how does evolution happen or what happens in the lagoon. So in this specific experiment, we're interested in evolving protein-protein interactions. And this is the concentration of phage with respect to time. Each one of these colors is a slightly different selection where we've tuned the selection to make it harder as we've progressed through each one of these arcs. So this went for about 3 weeks, and on average, variants that we pulled out from the very end had about 23 coding mutations. There's a lot of other mutations beyond that, many silent mutations, there's mutations in the phage genome, but for now all we really care about are the ones that are happening in the gene of interest. And if we then take all of those time points and sequence them, we can say something very interesting about how we went from the starting point all the way to the variants that were functional at the very end. And we can potentially even describe how the mutations were accumulated and how they were lost, and maybe that even tells us something about what mutations were important or ones that were not. The problem with this is 23 mutations is a lot to kind of decouple, in that combinatorially that number is huge if we're trying to figure out what mutations are responsible for the activity versus what mutations are hitchhikers, because we're introducing mutations non-specifically through the entire gene of interest, that sometimes maybe you get a mutation that's really important, but alongside that you get a mutation that's not detrimental to activity. And trying to figure out which mutations are important versus ones that are purely hitchhiking is very useful moving forward, because maybe the hitchhiking ones in this context don't hurt you, but later on in other contexts, which is what we actually observed, is that they can be incredibly deleterious. So I'll tell you that many of these mutations in the context of PACE actually function just fine. When we take these proteins and assess them in the environment that we want to assess them later on, they're incredibly destabilized. So these variants, actually, if we pull them directly out of PACE and try to do something useful with them, non-functional. So now we have to do mutational dissection, because we know that within that space of 23 mutants, there's a combination that gives us the activity that we care about. And so,
And you'll synthetically recreate that.
exactly. And that's what we did. So we synthesized on the order of 300 variants that combinatorially tested some of these mutations and found that only 9 of the 23 were necessary and sufficient for this activity. And this is a huge, incredibly labor-intensive effort. And what we'd really like to do is incorporate models to be able to better inform how to pick those mutations. In this case, because it's a protein-protein interaction, essentially even docking type simulations would be very beneficial to inform what mutations would be important. Yeah?
Sorry, just because it's very interesting.
Yeah.
Are you getting the average sequence data? Are you just saying like the when you do these sequences of each time point?
Yeah.
Are you just doing like Sanger sequencing and getting? Are you doing actual next gen
Yeah,
sequencing
so-
and a representation of each
Yeah.
quote unquote haplotype?
So the way that we got this data, so the gene is relatively large, it's about 2.5 kb. And we're not only interested in the mutations exclusively, but also we want to look for things like epistatic interactions. This is problematic for a gene that size for high-throughput sequencing because you can't really do that. So what we ended up doing is sequencing the pools by both high-throughput sequencing short reads like Illumina, as well as PacBio for the longer reads. And so Illumina helps us inform the mutations that we care about that are happening during the course of this experiment, and then using the PacBio data, we can generate this type of analysis. There's still a problem here, which is that for your typical HTS run, the Q-scores of 40, which is about as good as it gets, is, I believe, 1 in 10,000, a variation in 10,000 genes. The library sizes are about 10 to the 8th, 10 to the 9th here. So you're missing a substantial amount of diversity. So we're only describing what we can describe with the limits of high-throughput sequencing.
But that's not an issue of like inaccuracy of high-throughput sequencing.
No.
It's an issue of the depth.
That's right.
In this context, you're just not getting most of
That's
what's
right.
there.
That's right. So all we're really saying are the important things, ultimately. Right.
Well, I guess it also depends on how quantitative your switch is, right?
Yes, and that's a huge design consideration in that we try to make sensors that operate across a huge linear range rather than being effectively digital, because that ends up helping us in selections. Okay, so I think I only have 2 more examples. This one's a little bit higher level. Phage are very interesting. I guess they're not alive, but the interaction that happens between them and the E. coli is relatively well understood. There's a lot of information that we have about how bacteriophage replicate, what happens when they infect, kinetics that regulate the processes that underlie the host pathogen interaction, that we can actually carry out these models but we manipulate things like the phage generation time to inform things, to recapitulate how well the selection works or how quickly it operates, and the dilution rate of the lagoon, which is a very rough metric for how stringent the selection is. And because we're not computational biologists, we've had to determine empirically that these things rarely have an effect on selection. And this is across a number of different schemes, across a number of different trials. But what you can see here is that all this information is borne out of 4 equations, relatively straightforward way, computationally. And so I think using mechanisms that employ information that we already have about this interaction to be able to inform how we regulate things like positive or negative selection, how we tune the selection and stringency, which is, again, currently an art that we basically just go in, tune something a little bit, see what happens, and iterate this process. How we regulate the flow rates and how much time, this is actually one of the bigger issues, how much time we devote to actually doing the evolution. It turns out that variants arise relatively quickly to find the activity of interest, and the remaining time ends up being kind of like akin to maturation of antibodies, in that the starting point is found and then it gets slightly better. But we don't really need to be able to evolve it for 5 days if we can evolve it for 1 day and reach the same endpoint. And lastly, one system that I think would be really interesting to have is a slightly decoupled phage paste system. So the way that we have it organized now is there's a number of different phage genes. There's your favorite gene. It infects the cell and is regulated by these mechanisms and then undergoes evolution. But one issue is that the mutagenesis plasmid not only mutates your gene, it mutates everything else. And on average, half of every mutation, or half of all mutations that are made to the phage genome are actually deleterious. There's a huge reduction in library size, and we'd love to be able to obviate that. And because we know so much about M13 bacteriophage, what we can do is now generate these so-called helper plasmids, which provide all of the phage proteins in trans. And now our phage only encodes the thing that we're trying to evolve. So now all the mutations are accumulating in the gene of interest, and now we have substantially higher coverage of our library.
What's the helper plasmid?
So basically, this operates instead of like a virus, it operates like a plasmid and provides the phage proteins that are required for things like replication, construction of the viral capsid, all of those things. There's a problem with this approach, though, which is that the dosing of the phage proteins, when they come on, when they go away, all of this is very, very important to the efficiency of this whole process. And so the nice thing about M13 bacteriophage is we have just about the most information about how it functions, more than anything else that probably exists. So we know all of the proteins that are made, exactly how many proteins are made, when those proteins are made. All of these things that kind of beg a model to be constructed to define how to best construct a system where all of these things are turned on at specific times, at specific concentrations, to better generate a system that kind of operates like this. So that's kind of a brief summary of how both we've constructed the technique and also problems that we've seen along the way, and potentially solutions that we can use to address some of the schemes and data that I've showed. So I'll end with a bunch of acknowledgments. These are a lot of people that did a lot of the work that I presented. The vast majority of it was actually not my work. But the initial development was done by Kevin and Jake. And then for the different projects I've highlighted here, people that actually did the work. Obviously, I'd like to thank David for giving me the opportunity to work on PACE in his lab. And since I've left, I've actually transitioned to using PACE to potentially even understand more complex biological phenomena. So now we're using PACE in my lab to be able to understand antibiotic resistance, specifically antibiotics that target the ribosome. And so if we can understand that much better, we can theoretically design better antibiotics, or maybe combinations of antibiotics that now limit the rise of resistance, or even say something about how the ribosome came to be and what is necessary for translation, all informed by directed evolution in PACE. So with that, I'll end, and I'm happy to answer any more questions.